in the chat box as well, right? So you can join by that one. Just 1 second, where is chat box here? Yeah, I am sending one link in the chat box, right? So just let me know, can you see there, these. mm, can you see in the chat box? I have shared one link. Not working? Okay. No, no, I have not maybe started yet, but let me let me share my screen there, so then you confirm. Now, can you see in that link? Can anyone confirm? Can you see that? Can anyone confirm it? The link which I have shared in the chat box. Can you see my slides there? Hello. Link is not working. Let me share it again. Again, sorry for your. I am just killing your time as well. Uh, I am so sorry for that. I can understand that everyone's time is precious, you know. So I am sharing it again. Now you can check. Yes. Now can you see? Okay, everyone can see there. Okay, great. Now you can see it. Great, great, great. Okay, let me share the students who are not able to join by that one. Let me share with them as well. You know, I thank God it's working. Let me see what link I have shared with all of you. Copy link address. Yeah, because many are saying they are, particularly they are interested to join. Very difficult task it is. Hello. Hello. Yes, ma'am. Hi, Ghazala this side. Actually, I'm trying to join the SLR workshop. Yes, ma'am. But the link is not I'm trying to enter, but someone need to enter me into that. Ma'am, actually, what happened? That limit has been crossed. So, what now I am doing? I am sending you the. You just WhatsApp me. I am sending you the one link of the YouTube, so you can see in the YouTube as well. Okay, 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 ma'am. I am sharing that link. Shared with one person else. Yes, yes, ma'am. Sure. I already messaged her actually. Okay, ma'am. Okay, thank you. Okay. So we are discussing about, I hope my screen is visible on YouTube, and I am audible too, right? Yes, yes. It's up to you because in that one you can ask the question as well. I think in YouTube you can put your question in the chat box. But here you can, if you have, here you can't actually. You can see on the YouTube screen, but the questions you can ask here. No, let me. No, no, video is not restricted. Let me see. Let me check, sir. But video is not restricted, I think so. Same. Everyone is facing this issue. Video is restricted. So you are able to see on the YouTube my first slide, title slide, SLR and meta analysis. Okay, let me, sir, see the setting. Maybe in setting there is maybe something. Why it's restricted, I don't know, but let me check. Okay, maybe, sir, you have to be log in through your email ID, maybe. Any other one who is facing same issue? Can we start? If okay, I think for someone it's not working like that they are saying. So I am putting one more thing is so I am putting just this is live, and particularly in this one I am putting another link in the chat box. I hope it will be open, and in that one you will go to the live sessions, so it will work out. I have again put it in the chat box, right? Can anyone confirm it? I hope I am audible as well. Okay, great. And you can see the first slide on YouTube as well, right? So I think we can go ahead, right? So particularly, so workshop is related to your SLR and meta analysis, right? And as I said, SLR is a systematic literature review. Means so you have to be do the literature in a systematic way. So what is the systematic way that I will try to explain you how to do the systematic literature review in a systematic way, right? So going to start with the systematic literature review. Just 1 second. I think some are why we are facing this problem. So particularly, Okay, sir. Okay. But I think I am unable to share, sir, in both the things actually. In You can ask your queries in the Google Meet, but screen you can see on the Google this one YouTube. Yeah. So we are discussing about the what are the skill which is required, and that already we have covered. Means you should have these technical skill, then you can become a good expert in this particular field. So, particularly, we are going to start with what are the different types of review, which you have to take care. So, as I said, narrative review are something maybe many of you have written in your life as well, and many of you have seen. And how to differentiate between narrative review and systematic review is particularly you can differentiate. If you go through the article, you will find that if there is no methodology section, no result section, no discussion, so you will get a sense that this is a narrative review, this is not the systematic review. If you have to be differentiate between these two, so difference between these two is, if you see the systematic literature review, you will find that in systematic literature review, in method there is a methodology, just like a research articles, there is a introduction sections, methodology is there, so results is there, discussion is there, conclusion is there, so you will get to identify it's a systematic literature review, right? Then there is a another type of review which we talk about is diagnostic test accuracy review. If you are working in a diagnostic or these kind of things, so you can learn about diagnostic test accuracy review. There is another review which you might heard about, living review. Living review is something which are living का मतलब क्या होता है, जिंदा रहना. So these are the review which are updated time to time. These kind of review we call living review. Then there is a particular review that we call scoping review. You might heard about scoping review. Scoping review is something you are doing when you don't have a clear idea about the literatures or particular topic. You don't know whether, means there your questions are very broad, your questions are not specified, research questions are not specified. So in that case, you have to go with the scoping review instead of systematic literature review. So again, all things will depend upon your research questions. You know that your research questions, you knows about what is the populations which I am looking for, what are the interventions, what is comparator, what is outcome, what is study design, what is time frame. So you go with the systematic literature review. But if this particular if your research questions are not defined, your population is not defined, your intervention is not that much defined, you are going to take care whole class, in that case you have to be you can go with the scoping review. So scoping review is particularly we are doing when our research questions are broad, right? So systematic literature review. What is systematic literature review? It's a structured, transparent, reproducible method of identifying, evaluating, synthesizing all the available research evidence on a specific topic or research questions. Again, I am repeating, specific research questions or specific topics should be defined in your mind. So what should be defined? Before doing the systematic literature review, your research question should be clear. For example, I want to see what is the role of particular interventions in particular patients, and these are my outcome parameters. And this is nowadays we are also asking the researcher to define your research design as well. Research design means research article so which you are looking for. You can't mix RCT, randomized controlled clinical trial, with crossover designs, or so that we call observational studies. If you are doing this, you will get in the troubles. Expert will comment on your systematic literature review why you have combined two different research design. One, you have combined the data of randomized control trial with case control studies or cohort studies, that are the observational studies. So don't combine these two different study design. You have to be define your research design which you are looking for. and it should be according to your research question. For example, I want to see what is the efficacy and safety of XYZ drug in particular patients. Nowadays, you might heard about GLP-1 agonist, right? Everyone is talking about semaglutide. For example, our team is doing one SLR and meta-analysis on semaglutide or GLP-1 receptor agonist, and we want to check its safety. So our research question is defined. Our research question are defined. What actually? I think just 1 second. Our question is defined. My treatment is defined. I am looking for a GLP-1 agonist. In GLP-1 agonist, which drugs? For example, I am looking for semaglutide and liraglutide only. Then what is the comparator? Then what is the outcome and study design? We are looking only for the randomized controlled clinical trial, and randomized controlled clinical trials are the gold standard. Now question is why? Can anyone answer it? Yes, anyone can answer it. I hope I am audible. Why the randomized controlled clinical trials be considered as a gold standard when we talk about the evidence? Anyone? Yes. Yes, very nice. Very correct answer. Because chances of bias is less. Chances of bias is less because you are doing the randomizations, which reduce your selection bias. You doing the blinding, which again decrease your selection bias, right? So what I am saying is, if you are planning for SLR and meta-analysis, please, define your research design as well. Which kind of studies you are looking for? I am not saying we are not doing the SLR and meta-analysis of, observational studies. We are doing it. What observational studies? If we talk about level of evidence, we are giving low priority for the observational studies. High priority we are giving for the RCTs. And as sir has said, that is the one of the reason, right? Their biasness is less in case of RCTs. So systematic literature review again means your review questions should be defined, your search strategy should be defined, your selection process, strength of evidence, analysis, and the synthesis. And another thing which I also want to just emphasize that sometime what happens, students comes to us and they are saying, sir, we want to do one meta analysis on this XYZ topic. And when they have completed the SLR, systematic literature review, now you understand that okay, on this topic we can't do the meta analysis, we can restrict ourselves up to SLR. Now student feel bad. I asked the sir to do the meta analysis. Now sir is saying to me, restrict yourself up to SLR. So again, means it doesn't means every SLR has to be converted into a meta-analysis. It depends upon what kind of data you are getting from the literature. So once you will complete your systematic literature review, you will decide, means depending upon what kind of data. There is a lot of issues, when you do it practically it looks like simple, but when do it practically is many researchers, some researcher are putting their outcome in terms of mean plus minus SD, another one is putting in mean plus minus SCM, another one is putting in IQR, another one is putting in another terms. So is it easy to pool all the data in a uniform format? It's not that much easy work. So depending upon what kind of data we are getting from the SLR, we have to decide whether we have to go for the meta-analysis or not. It's not like that you thought we will do meta-analysis, so I will do this meta-analysis, right? It's not like that. Still, good SLR is published in a very good journals, and that are good SLR is also the finding for the policy making as well. Got it? I hope you got my points. What I want to convey, again I am repeating, I want to convey that means you are passionate about meta analysis, that's fine, but again it depends upon what kind of data we are getting after completion of SLR, then you decide. And if you feel no, means for statistical analysis this data is not enough, heterogeneity is too much, otherwise if you are going for a publications in the form of SLR, it's easy to publish as compared to meta analysis which you have done in a inappropriate way. There are a lot of heterogeneity, everything is there, data is not means up to mark, number of studies too less. So reviewer will put the questions on your meta analysis, and what if you restrict yourself up to SLR, it will be publishable. So that I think researcher has to be understands where you have to stop. Then next thing is meta analysis. You all might have heard about meta analysis, right? Meta analysis, there is a word analysis. So when we talk about analysis, what is required to do the analysis? We need data, right? Without data, how we can do the analysis? We can't do, right? So to do the means meta analysis is provide the clarity among the published studies. So we will discuss in upcoming slide when you have to plan for the meta analysis, right? It's not means what are the good topic for the meta analysis. So it's a quantitative approach. When we talk about meta analysis, we talk about quantitative. It's a quantitative approach. SLR is qualitative approach, but after doing the SLR only you can do the meta analysis, right? So it's a quantitative approach for systematically, again word systematically is mentioned, combining result of previous research, right? Previous research to arrive at the conclusion of the body of the research. And we are calculating pooled estimate that we will discuss tomorrow as well, how to calculate the pooled estimate and how to select the pooled estimate, right? So that we will discuss in detail, right? I hope it's going fine. So to simply understand, these are the pictures. Just I think in 4 5 year back, I have attended one workshop on SLR Meta, and they have to easy explain to the audience. They have put it such kind of picture. So I have taken this picture from the Google Images. So it's just like you can see in this picture. What is there in this picture? There is a net, and this net has catched the particular feces, right? So meta analysis or SLR is like this only. In C, there is a lot of pieces. In literature, in PubMed, there is a million of research papers are there. So you have to put the net to catch particular pieces. So here, what net? Net is what? Net is your search strategies which you are using to catch particular literature from the databases. So if you see, net is the your search strategies which you are putting, that I want to include only these pieces. Then after collecting the fishes, what you are doing, what means fish, not you, means fisherman is doing, they are doing the assessment of these fishes in terms of quality. Okay, if I am collected these many fishes by putting this net, right? So how many of them are of good qualities, how many whatever the fishes of bad quality, it will be separated out. Yeah, these are the medium, these are the these categories, right? In similar way, we are also doing in SLR meta analysis. We are making the search strategies that is just like a net. You are putting in the ocean is your databases which contains the literature, right? Number of literature is there. Are you getting my point? So after getting these literatures, we have to do the quality assessments as well. We have to do the quality assessment, right? Without quality assessments, again there is a risk of bias. There is all assessments you have to be do. Just 1 second. I think many are trying to admit as well. Still far there is any questions? So far there is anyone have any questions? I will see the participants who are unable to join through the Google Meet and watching on YouTube. They can put their queries in the YouTube chat box as well. I'm just opening my this one through the mobile as well. I will take the queries from there as well. Yes, let me know any questions so far. So it's just like a pieces. You are putting the net. Net is your search strategies. That's why now by seeing these pictures, what is the most important step in the SLR meta analysis? If your net is not good, अगर नेट में होल है, नेट में आपका ये मछली पकड़ी नहीं जाएगी, तो वो तो निकल जाएगी ना. Are you getting my point? So this net is very important. So net is your search strategies. फिर After that, when you are collecting it, then you are doing the segregation, which of good quality, which of low qualities. And again, it's a garbage in, garbage out, because you are making the conclusion out of the literature which is already published, research studies which are already published. So if you are making the conclusion by including lot of studies which of low qualities, so who will believe on your results, who will believe on your conclusion? The person, the expert or physicians will always say, okay, this is your conclusion, but what is the basis of this conclusion? So you will say, these are the studies which I have included, right? So particularly, next thing is, what is the difference between meta-analysis and systematic literature review? So you can see that bigger circle is systematic review, smaller circle is meta-analysis, right? So why this bigger circle is systematic review? The reason is because every systematic review may or may not, every systematic review can't be converted into meta-analysis, but for every meta-analysis you have to do the systematic review. I hope it's clear. So if you see the health research pyramid and categories, as well as we are discussing about, if you see at the lower priorities, we are putting animal studies, lower evidence, then there is a case report, case series, then there is a cross-sectional studies, then there is a case control studies, then there is a cohort studies. I think someone is asking for admit. Yeah, Sweta, you want to ask something? So health research pyramid is like this, and particularly, RCT is getting the maximum priorities, and then there is a meta-analysis, and on the basis of meta-analysis and systematic review, we can make the clinical practice guidelines. Now, next question is when it's required. When it's required is, when there is a confusion among the physicians, then you have to be think of the SLR and meta-analysis. Say, as I said, you can't pick up such kind of topics where clinical evidence is already clear. If we talk about interventional clinical trials, so if there is everything is for example, if I ask you, if you are planning to do the SLR and meta-analysis with respect to use of aspirin in myocardial infarction patient, is it a good topic for the meta-analysis? Can anyone answer it? Yes. Is it a good topic to do the SLR and meta-analysis? If I say that use of aspirin, I want to see efficacy of the aspirin in myocardial infarction patients. So is it a good topic? Okay, good. Any other one? Anyone want to add? If, for example, if someone want to see what is the effect of statins in hypercholesteremia, in on the lipid level, is it a good topic for the meta-analysis? Yes or no? Why? Yes, 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 perfectly, perfectly fine. You are right. You are right. Perfectly fine, because it's already known that sir, this particular is means we have a enough evidence which shows that statins reducing the blood cholesterol levels, right? So that are not a very good topic for meta-analysis and systematic review. That's why on screen you can see that it mentions when there is a confusions in the clinical practice. Doctor are also confused whether we have to use it or not to use it, because some set of studies saying it's effective, some set of studies saying it's not that much effective, its safety concern is there, some study are saying effect is there but it's a non-significant. So such type of topics are very good, right, to conduct these SLR and meta-analysis. So selection of topic is very important. As I said, selection of topic for SLR and meta-analysis it should be the conflicting evidence. Pick up such kind of topic when there is a conflicting evidence, right? And as I said, there is a some topic which are important to make the policies, like with respect to antibiotic guideline of India. Recently ICMR has given the funding for the researcher to do SLR and meta analysis. Let me see if I can admit. So whenever there is a confusions, so in that case you have to be pick up such kind of topics for the SLR and meta analysis. Ongoing scientific argument is there, right? Check availability of the scientific data as well. Sometime what happens, your topic is very good, your topic is very good, your research question is good, there is a conflicting evidence, this research question is for the policy, it's very important, ongoing scientific argument is there. But when you do preliminary research, you will find that there is only one or two studies are there. So is it a good topic? Are you getting my point? So you have to check before that one. You have to check availability of the this one as well. Particularly the availability of the papers as well. Means how many studies are there. Next is Prisma. I hope many of you heard about Prisma 2020 guidelines. So Prisma, you have to be follow when you are planning for SLR meta-analysis. And Prisma stands for Preferred Reporting Items for Systematic Review and Meta-analysis. It's first published in 2009 and revised in 2020. So sometime what researchers are doing, they are doing the mistakes when they are using the Prisma. You have to use the recent Prisma. Okay. So you have to be use recent Prisma. So recent Prisma is 2020. So what is the difference between these 2020 and 2009? The expert can easily identify which Prisma flow chart you are using in your systematic literature review. So you have to be careful when you are using it. Prisma is made up of checklist and flow diagram. Checklist set out of 27 items, and that must be reported in every review. And if you go through the Prisma, you will find that you have to describe the result of the search selection process, number of record identified in the search, and so on. And you have to present it all these in the flow diagram. There are two things: one is flow diagram, another is checklist, right? So checklist is something. This is a Prisma 2020 checklist. So this checklist you have to be fill and put as a supplementary file. So this is important, when you are submitting your SLR or meta analysis, you have to fill the checklist. So this checklist is available. If you do in a Google Prisma, you will find that. So you will find, go to the Prisma websites. In Prisma website, you will find there is a two document. One is checklist, another one is flow diagram. Sometime what the researchers are doing, they are creating flow diagram by their self as well. Someone is creating it in the paint, someone is creating in the drawing tools or any other tools. They are creating by themselves. There is no need to create this Prisma flow diagram. You have to be flow diagram, you will automatically get in the website. Just download it and fill it. Similarly, you will get the checklist as well. So this is a checklist. So in checklist, once your SLR or meta is completed, after that you have to fill this checklist. And in this checklist, you have to be mentions, okay, in title, identify the report as a systematic review. You have to mention where it's located. Abstract, in abstract, where it's located. Rationale, objective, eligibility criteria, methods. When you completed your SLR meta-analysis, after that, you have to fill this checklist and attach it as a supplementary file. Got it? Any questions so far? Anyone has any questions? So flow diagram, this is the flow diagram. So again, I am repeating, flow diagram, there is no need to create the flow diagram. You just download it from the Prisma websites and fill the number. If you see it carefully, there is a n is equal to, n is equal to. So how many studies you are get from the database, how many studies you are getting from the registries, how many studies you have excluded, how many studies you have included. So these number you have to be filled. There is no need to create this flow diagram by yourself. It's available on the Prisma website. And similarly, there are two things, as I said, Prisma checklist and Prisma flow diagram. So you have to mention how many studies at initial search, how many studies when you have done the first pass screening, second pass screening, and finally how many studies from which you have extracted out the data, right? There is a, if you are going for a Prisma scoping review, even though we are not discussing about scoping review, but if you are, then you have to Prisma extension for the scoping review is there, and it's published in 2018. And if you see that the checklist contains 20 essential reporting item and 2 optional item when you are completing a scoping review. Again, I am clearing all of you, scoping review you have to be planned when your research questions are not defined. Sometimes it happens, okay sir, you are saying to do the systematic literature review, your population should be defined, your intervention should be defined, your comparator should be defined, your outcome should be defined. But if it's not defined, but you say sir, but अभी तो मेरे दिमाग में क्लियर नहीं है, मैं तो ये देखना चाहता हूँ कि लिटरेचर में है क्या एक्चुअली. So in that case you have to go for the scoping review instead of SLR, systematic literature review को छोड़ के आपको scoping review पे जाना चाहिए. Now, first and most important part, when your research questions are defined, right? Now you knows, okay, this is my topic for which I want to do SLR meta analysis. So what next I have to do? So next is you have to do SLR protocol development. You have to develop the protocol development is very important. So many times we are doing the mistakes, we are completing our SLR and meta analysis, and still we have not completed our protocol, and we have not submitted for the registrations. So that is the mistake that you have to avoid. If any one of you are planning for a SLR meta analysis, first write your protocol. When your research question are defined, next step is write your protocol. So protocol sections are there. I hope everyone of you knows about protocol, where you have to be describe your objectives, eligibility criteria, which studies you are going to include. As I said, during the research question, it should be clear in your mind what type of design of the studies you are going to include. Whether you are going to include only the RCTs, whether you are going to include observational studies, whether you are going to include animal experimentations as well. You can include animal experimentation as well, but it should be separate. You can't combine animal experiments with the human studies. Are you getting my point? So otherwise, experts will arise the questions how you have combined two different types of studies. So if you are, preclinical researcher, so you can do the SLR, but in that one you do the, so you can make your inclusion criteria that I am going to include only the animal experimentation studies on this topic, and make your exclusion criteria clear. Other than that, I am going to exclude. You have to define your time window as well. Means, okay, because literature is something which is going to updated on regular basis, right? Per day it's updated. Today if you are doing the PubMed search on particular topic, maybe you are getting means, for example, 10,000 article. Tomorrow if you are doing it, the number will be changed, right? So you have to define your time window as well when you are doing such kind of studies. Search strategies, you have to define quality assessment, how you are going to assess the qualities, method analysis, and representation of the results. And then also you have to describe in the protocol plan for the minimization of the bias, how you are going to minimize the bias. If there is a heterogeneity, how to address the heterogeneities, which factors could affect the result of the studies as well. So SLR, you have to go for the registrations, and for the registrations, this is the one of the well known, means many researchers who are working on SLR and meta analysis, this is a well known registers where you can register your protocol of the SLR. So you just go through, you just type in a Google's Prospero, it's freely available. You just click on the join, you can create your login ID and passwords, then do the login, and after login you can see that it will open like this, my Prosperos. So my Prospero, you can see that here is the option register your review now. Just click on register your review now. So it will ask these type of questions. So here you can see that it will ask you whether you are going to include only the animal studies, whether you are going to include your study participants or pupils. So if you are your SLR or meta analysis is on human, so click on these options. If it's animal, so click on this option. Then after clicking these options, you will get like this. Well, will you register record in English? Will is this a scoping review? Another thing is, scoping review is you will not get the registration from the Prospero. For scoping review, there is a different one. I will show you in upcoming slides. So you have to register scoping review. If you are planning for a scoping review, registration is different. Register is there. But if you are clicking here, is this a scoping literature review or mapping review? Yes. If you are clicking it, it will, next question will not be appear, right? So if it's not a scoping review, then it will be registered. I hope it's clear. Yes, ma'am. Any question? So, have you started your reviews? So it will be asked. If no, then it will be open like this, right? So you can see that review titles, particular actual start date, anticipated date, your inclusion criteria, exclusion criteria, risk of bias assessments, all these. So all these informations you have to fill. Are you getting my point? Anyone has any question up to this? I hope my slides are visible. Okay, so after that, you will get the registration number. Once you have to submit it, you will get the registration number like this on your email, right? So you can see that this kind of registration number you will find, and that registration number you have to mention in the methodology sections of the systematic literature review. When you are writing systematic literature review or meta-analysis, in the methodology sections, you have to mention the registration number. This is this protocol of this particular study is registered with Prospero. This is the number. Okay, ma'am, I think you can see the screen on this one live streaming on YouTube only, and here you can ask the questions. I think someone is asking what is meant by supplementary files. Supplementary, we will take all these questions. You put in the chat box, start putting in the chat box. I will take after completion of this introduction to SLR meta analysis. Okay. So there is a open science framework is the particular database register where you can register the scoping review, where you can register the scoping review. As I said, if you are planning to register scoping review in the Prospero, it will not register. Prospero, there is a one question it will ask you whether it is a scoping review. If you are clicking on yes, it will not move further. So what does it mean? It means that Prospero is not doing the registrations for the scoping review. Again, I am clearing scoping review, but SLR it will be registered. There are many SLR which are registered in the Prospero. The in policy is another register where you can register your protocol. This is the open science framework, as I said, where you can register your protocol of scoping review. Another thing is, meantime, there is a confusion among the researcher also arise because in systematic literature review or meta-analysis, most of the researcher are dealing with the human data, right? So whether there is a need to get the human ethics approval to do the SLR or meta-analysis, so ICMR has given one statements or particular amendment they have done and mentioned that whether human ethics committee approval is required to do the SLR or not. So what they have mentioned clearly that if your systematic literature review or meta-analysis protocol is following the standard guidelines or procedures, or your registration is done in the register in the international or standard database or registry, so it does not require submission to ethics committee for the clearance. There is no need to submit your protocol to the ethics committee for the clearance. Got it? But if it's not, you are not following, you are not doing the registrations, then you have to. Are you getting my point? So this is in 2024 they have done and they clear about it that if your systematic literature review or meta-analysis is following the standard guidelines, procedures, including prospective registration in a standard database registry, so it does not require submission to ethics committee for the clearance. Got it? So that's why the important thing is, as I am again repeating, whenever you are planning for a SLR meta-analysis, first write the protocol and submit it for the registrations. Got it? Important point, as I said, methodology for the systematic review should be structured, transparent, reproducible, which will provide reliable finding, make a valid conclusion out of it. Researchers should be clear about the scope, eligibility criteria, which studies you are going to include, which studies you are going to exclude. Of course, some of the studies you have to always exclude, like review articles. Review article, you, unless and until if you are doing systematic review of already published meta-analysis, so in that case you can include. But in most of the SLR meta-analysis, if you see the exclusion criteria, we are not going to include the review article, because we want to extract out the research data on that particular topic, right? Searching of all the possible databases, issue of bias and analysis of the included studies, and always involve a team. If you are planning for SLR meta-analysis, involve a team. You need at least two independent researcher to do the screening of the studies. It's mentioned in the guidelines. Two independent researcher for the screening, and they should be blind to each other, and then only you can resolve the disagreement. So third expert should be there. Then expert from the field, in which area you are planning to do the SLR and meta analysis. So that will help to design your data extraction sheet, validation of data extraction sheet. For example, you are working in the diabetes, so you try to include someone who has worked in the diabetes since long time, or someone from the clinical point of view. You can include the clinicians who are dealing with the patients of the diabetes, endocrinologist you can consider, and one biostatistician you can consider. So it's always advisable to include the team when you are planning for a good SLR and meta analysis. So team should be very strong and team should be diverse. Means one biostatistician, another one is clinician. If clinician, I can understand that clinician don't have that much time. But if someone who is working in that particular area since long time, so they can give their input during the designing and validations of your SLR and meta analysis. So predefined protocol, what, why, and how, that should be defined. Clearly formulated questions, research questions should be there. Like when we talk about SLR meta, we talk about PICOs. You might heard about PICOs, right? PICO is, so PICOs is something where you have to define your populations or patients. I is the interventions, what interventions you are giving, comparators, outcome, study design, time frame, right? So population, so for example, so I want to see safety and efficacy of bile acids in MASLD patients. So my population is MASLD patients, my intervention is bile acid, my comparator is non bile acids, my outcome is particular, what are the parameters with respect to fatty livers. My study design is I am going to include only the RCTs. My time frame is I want to include studies, means so for 1 years or 2 years. So all these should be, your research questions should be formulated in the PICO formats. Comprehensive search strategies. Your strategies should be like that. It will it will not miss out any important studies. So it should be comprehensive, and also it should not be like very much generalized, so that you are getting so many articles, right? And practically it's not possible to screen so many articles, because your search strategies you have not make it specific. It should be specific, but also enough sensitive to capture all the literature with respect to your research question. So search strategies also play important role. That's why whenever paper comes to us for the review, we always see the search strategies, what search strategies the researcher have used for these research questions, whether they have used proper mesh terms or boolean operator combinations they have used correctly or not. If there is a mistake, so whole process will be go in the vain, right? Transparent inclusion and exclusion criteria, quality and bias assessments you have to do that, we will discuss. And then you have to do the data extraction in the data extraction sheet, and after getting the data in the data extraction sheet, now you can plan for the meta analysis. Now you can think of, okay, I have enough studies, enough data, heterogeneity is less. Now I can go for a meta-analysis. Now I can analyze this data by generating the forest plots, by funnel plots, right? I hope it's clear. Any questions so far? So I think we can take those few questions. Yes, sir. Yes, sir. Yes, sir. Sir, we have to be means trial number is not required, but we have to be in the protocol. We have to mention our research questions, inclusion exclusion criteria, and particularly how, if you are planning for a meta analysis, how you are going to analyze it, and submit it to the Prosperos, and you will get the email. Now Prospero is very quick. Nowadays you will get the response within 1 or 2 days. Earlier we are getting the response after 15 or 20 days. So immediately you will get the publications in the Prospero. No, 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 sir. Trial number is not required. Trial number is not required because Prospero is the protocol. So the trial number is not required because after this protocol registrations, you are starting your screening process. So you don't know the trial number which trial is going to be included, right? Any other questions? Anyone have any queries with respect to SLR metas? I'm happy to take it, or I can take in live streaming. There are, I think, so many questions. So one by one, I will try to take less risk of bias. Okay, that is, I think I have asked the questions. Yes, you are audible. Okay, sir. Because of relationship is already well established. Yeah, that is the question which I have asked. What kind of topics you have to be include in the SLR? Means you have to go for the SLR meta-analysis, right? Uh, then meta-analysis is used to combine the result from multiple study to obtain one strong and reliable conclusion. It is useful. Sorry. It is useful when study results are different or confusing. Right, you are right. Can do meta-analysis. There are a multiple option available and confusion among the efficiency. Yeah, means if there is a confusion among the efficacy, I hope you are talking about efficacies. So you can go for the meta-analysis. So what is meant by supplementary file? Supplementary file is, so when you are going for a publications, so in that one you can putting such kind of materials which supporting your results, but that is you can't include in the main file, right? Because if you are putting all the things in the main file, your article will become too big. It because journal has a limited space for the articles, right? So in such cases, what journal is asking, whatever the material which is important and supporting your findings, you can put in the supplementary file. So Prisma checklist is something which ensure that you have followed the guidelines, you have mentions, you have your writing has been done according to the guidelines. So that Prisma checklist you have to fill and put in the supplementary files when you are putting in the journals. So put it in the supplementary file, not in the main file. So in SLR meta-analysis, you might have seen that there is not a, in main articles. Means I am 100% sure that no one of you have seen Prisma checklist in the main articles. It will be included in the supplementary file, right? Any questions? Any other questions in the chat box? Okay, in time in Google, this one, Google Meet. Any questions? Yes, go ahead, ma'am. Yeah. Mm-hmm. Sorry, can you repeat the question? Yes, yes. Minimum we are, yeah, I got your question, ma'am. Minimum is 10 studies we are considering. Means minimum should be the 10 studies should be there in the literature, right? Even for the meta-analysis also, so when you are going for a publication bias assessment by the qualitative assessment by the funnel plot, so minimum 10 studies should be there, right? So that's why I said, means sometime your topic is very good, conflicting evidence very good, but evidence is not there, means only 1 or 2 studies. So 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 maybe after 1 year, when more evidence will be generated, that time it will be the good topic, right? Any other questions? Any other questions? So I hope you understand the important points. You can put in the YouTube chat box as well if someone is watching. If anyone is watching, they can put their query in the chat box as well. So summary, whatever we have discussed so far in last 40 or 50 minutes, if you ask me, sir, summarize it, what are the important points? So I just summarize it. First thing is, when you are planning for a SLR meta analysis, first thing is you have to be, your research questions should be very much clear. So it has been observed that many times our research questions are not clear. So when students are doing the screening, first pass screening, it's very difficult because students is not clear about which article, because in screening you have to decide which article has to be included, which article has to be excluded. If research question is not clear in the mind of the students, so they will do the mistakes in the screening. So your so screening will be affected by that one. So another thing is, second thing is, when your research questions are defined, everything, uh, you are thinking of, okay, at this stage I am not clear about which population I am going to include or which interventions I am not that much clear about. My topic is, this is the areas, and particular I want to do these things, but my question is not, I am not able to convert into the pcos. So in that case, go for the scoping review, not for the SLR. Scoping review are also published in Lancet or JAMA as well. If you go through Lancet, JAMA, these are the prestigious journal in the medical field. So you will find that these journals are also, so these articles are published in these one, means in these good journals. These articles are published. Scoping review I am talking about. Another thing is, when you have cleared your research question, second thing is, write the protocol. Many time, many students, faculties, they are missing this point, and that is very important. As we discussed in animal studies, whether you are, you can touch the animals without approval of animal ethics committee. No, right? Can you means can you do the human studies without the approval of human ethics committee? No. So you have to submit the protocol, then only you can do it, right? So similar way, you write the protocol, submit it, and get the registration number and mention it, this protocol is registered with this one. Got it? Any questions apart from this? And next we will discuss about the search strategies as well, how to make the effective search strategies in the PubMed according to your research question. In chat box there is any questions? No. PPT not visible. Yeah, that I am very sorry, because I am not expecting that many will join, because many times we have observed that, when we are conducting webinars or all these things, means participants are registering, number is too much, but so when actual webinar started, no one joined. When means very few joined, particularly in weekend. So I am expecting less than 100, truly speaking, but it's more than 100, and many are putting messages and call to me, and I am alone to handle this. So due to that, there is a initially glitch, so I make it on live on YouTube. So I hope now it's clear to every one of you. But of course, means I am not able to screen in Google Meet this screen as well. You can put the question here, but screen you can see on the YouTube. Is it okay? Any questions, or we can move ahead? Yes, anyone has any questions? Hello, my screen is visible. Due to network issue, video is my top side. Can you repeat, sir? Why the business? Video started. Okay, great. So, is my screen is visible now? Steps in meta-analysis. Can anyone confirm it? Screen is visible. Okay. Let me see. Now we are in YouTube, not on Google Meet, because same time we can't, share on both. Actually, that is the problem. Not on YouTube yet. Okay, let me share it again. I'm sharing it again. Please confirm. This is the online problem actually. Now, now I think visible on YouTube, right? Can anyone confirm? Steps in meta-analysis, right? So now, if anyone of you are planning to do the SLR or meta-analysis, so what are the steps? So so what steps you have to take? So first step is formulate your research question that we already discussed. First thing is your questions should be for your research questions should be formulated in the PICO format. Again, PICO is P is for the populations, or you can call it patients, or interventions, comparator, and outcome, right? And nowadays, as I said, we are talking about study design as well, and identify your studies. Then step two is after your research questions you have break down into the PICO format. Then what next? आपने बोला सर I have the research questions and I have defined my PICO. So next what I have to do? Next thing is, of course, you have to write the protocol, and in protocol you have to define your inclusion criteria, exclusion criteria, all the things, right? So inclusion means which studies you want to keep. Again, it depends upon your research question. For example, your research question is on type 2 diabetes mellitus patients. So your inclusion should be, you are going to include only those patients who are of type 2 diabetes mellitus, not type 1. So it depends. Inclusion and exclusion criteria is depend upon your research questions. Are you getting my point? So which ones you have to throw out? Exclusion. So as I said, normally, in exclusion we are review articles, case reports, already published meta-analysis that we are going to exclude. Now, one question arises in my mind, and I just want to ask all of you is, for examples, your topic is very good, everything is very good, and number of studies also there in the databases. So my screen is visible. I hope my screen is visible, right? So it's moving as well. If I am going to the identification of the studies, and then you know yeah, I think it's working. Working on YouTube. So steps in meta-analysis. My question is, can anyone answer in the chat box? My question to all of you is, for examples, your research question is very good. You have done the preliminary search. You find that this is means enough studies are there, and we can go for the SLR and meta-analysis. But I am saying that still there is a one thing which can prevent you to do the SLR meta-analysis on that particular topic. So what is that one thing which can prevent you to do the SLR meta-analysis? That is my question. Can anyone put in the chat box? अगेन इफ आई रिपीट इन हिंदी, मेरा क्वेश्चन ये है कि आपका टॉपिक बहुत अच्छा है, ठीक है, कन्फ्लिक्टिंग एविडेंस पे है, और उस स्टडी उसको आपने थोड़ा सा प्रीलिमिनरी सर्च करके भी देखा तो उसमें हैं स्टडीज हैं लिटरेचर में, फिर भी आपको एक चीज रोक सकती है उस एसएलआर और मेटा एनालिसिस को ना करने से। वो क्या एक चीज हो सकती है? कोई बताएगा चैट बॉक्स में? वो एक चीज क्या हो सकती है? एनीवन? कैन आई सी द चैट बॉक्स? यस, एनीवन? Slides, I think now it's working. Maybe it's already registered. Okay, someone is saying maybe it's already registered in Prosper. Okay, you are too close to answer. Preliminary study is not SLR because it's exploratory, non-systematic, lack structure, methodology and quality. Okay, so the answer to this question is, जो एक चीज आपको रोक सकती है वो वही रोक सकती है कि फॉर एग्जाम्पल योर टॉपिक इज वेरी गुड बट इट्स ऑलरेडी पब्लिश्ड, और अभी आपके पिछले महीने पब्लिश हुआ है वो टॉपिक, और वही टॉपिक आप उठा रहे हो. आर यू गेटिंग माई पॉइंट? So already, if it's already published, so how you can means you can do it. There is no one can prevent you to do it. But once you completed it, reviewer will ask you the question, expert will ask you question. This is already done, and no study has been added after that. So why you have done this exercise? It's already done. Are you getting my point? Why? So that's why when you are planning for SLR meta-analysis, also do the search in the literature, what whether already meta-analysis or SLR is published on this particular topic or not, right? If it's published, then next thing which you have to be say is in which month or year this particular SLR meta-analysis has been published on this particular topic. That also you have to be checked. I am not saying if article is already published, meta-analysis already published. For example, let's say case I am planning for a one topic, and when I go through a literature, I find that this meta-analysis is already published, but it's published in 2010. So it's published in 2010. So from 2010 to 2016, only so many years has been covered. Means completed. So now evidence can be changed. So still it's published, but there is a lot of gap. So in that gap, because for example in 2010 the researcher who has done it, they have included only 10 studies. Maybe that time only 10 studies are available, but now there are a 20, 30, 40 studies are there. So when you are doing the analysis, your conclusion can be different from that one as well. So that we called updated systematic literature review. Are you getting my point? Anyone who has still doubt? See, selection of topic and steps are very important. So step by step. Otherwise, these are the common mistakes. You are very good in the methodologist, you are very good in the, but you have not done these checkpoint, so you will be in the trouble. Whole exercise will go in the vain, and it will not be published in anywhere. Because if for example Lancet में और JAMA में already last month सेम टॉपिक पे अगर मेटा एनालिसिस पब्लिश हो रखी है, और उसके आपके एक मंथ बाद आफ्टर वन मंथ यू आर डूइंग इट एंड यू आर ट्राइंग टू पब्लिश इट, everyone will raise the questions, means because you will get equal number of studies whatever they are getting it, and maybe you will get less because you have a limitations to search in all the databases, because many databases are paid one as well, like Embase is paid one, right? So that's why that is very important to search in all the available, I mean sir, to search before starting the systematic literature or meta-analysis on particular topic. Do the search in the literature to check whether someone has already done it or not done. But again, as I said, don't worry if someone has done it, then just check in which year, what was their research question, is my questions are different, is this study is published in 10-year back or 5-year back, and after that there are new studies come out, and now my conclusion can be changed, or now my systematic literature review is going to be updated. So that's the strong point to go ahead. But if it's recently published, and so then you will be in the trouble, you can't defend it. Are you getting my point? Anyone has any questions up on this? These are the very important points. Yes, okay, thank you. So next thing is, once you are clear about your research questions, Now you have to be extract the data from the included studies. You have to do the first pass screening that we will discuss, second pass screening, and after that you have to be pull out the data in the data extraction sheet. Now on data extraction sheet also there are many researchers are doing the mistakes. You have to design your data extraction sheet according to your research questions. If you see in SLR meta analysis, there is a table 1. Table 1 is telling us about characteristics of included studies, and if you go through the characteristics of included studies, some column will be same for each and every SLR meta analysis, like total sample size, age, sex, right, your locations. All these are the same headings you will find in each and every meta analysis table 1, that we call characteristics of the included studies, type of design, phase of the trials, right, all these. After that your research question will be different. Means after that your column of the data extraction sheet will be different. It depends upon your research question. For example, my research question is to check the effect only on HB1AC levels and glucose level. So I have to be put HB1AC levels in control group, their values in mean plus minus SD, number of patients in control group, then in a treated group. Then I can apply the statistics. So next step is you have to pull out the data in the data extraction sheet and then apply the statistics on it, and then you can generate the forest plot. You can get to know that whether there is a significant differences or not, and you can do subgroup analysis as well as the sensitivity analysis, that we will discuss one by one, step by step. So first part we are going to discuss is identification of the studies. This is a very important part, you know, where you have to identify. Now your research questions are defined, everything is defined. Now where you have to be search, means where you have to identify your studies, right? So identification of the studies, it's always advisable as per the guidelines. You have to search minimum 2 databases. अब यहाँ पे क्वेश्चन अराइज होता है, मीन्स मैनी इंडियन रिसर्चर आर फेसिंग प्रॉब्लम्स to get the data from the paid databases. PubMed is freely available, that's fine. But see, if you go through the Embase is paid one. Are you getting my point? Embase is paid one. So in that case, how to deal with that? So you can deal with that. Just 1 second. So how to deal with that? You can collaborate with the researchers who has this access of these databases, right? So you can include that. Another thing is you can search for the trial register as well. CTRI or clinicaltrial.gov, you can search it. You can do the hand search as well. Go to the library, do the hand search, personal references, emails. Some article if it's not opening, you can put the email to the corresponding author as well, right? You can put the email to the corresponding authors and so on. Another thing is Google search and so on. So first thing is formulate your research questions. Formulate your research questions. Particularly you have to combine. You have to use the mesh terms that we call medical subject heading. Medical subject headings we have to be combined with the Boolean operator. What are the Boolean operators? Boolean operators are your and or. So you have to combine with that one, and you have to put the proper filters. For examples, you can see on your screen. Just 1 second, I think. So how to, for example, your research question is efficacy and safety of gapiron in the treatment of MDD. So how to set up the search strategies? So how to do it? If you are putting this one gapiron or MDD, it will include so many articles. Why it is so many articles? Anyone? If I am putting first search strategy is, I am putting my research question is role of gapiron in MDD. So if I am putting this gapiron or MDD, I will got so many articles, right? Why it is so? Because or is not the useful tool for this particular for this particular research questions. Are you getting my point? Any question on this? So you have to use and instead of or, right? Then your research means according to your research questions article will be come out. If I am using or, then it will pick it up so many articles. So PubMed, everyone of you are aware about PubMed. So I can show in the Google as well. Can anyone confirm it? Is my Google page is visible immediately? Systematic review, lack of access to paid databases are managed by using institutional access, open access article, and multiple free databases to avoid missing important studies. Yeah, but still, so you can't access the Embase. Okay. So in PubMed, you can click on the PubMed. Here you can see the advanced options, but before advanced, you can see that here is the option of the mesh database. Click on this mesh database. If anyone of you want to do this exercise, they can do with me. Open PubMed in your laptop. PubMed open कर लीजिए. You open the PubMed, right? Again, I am repeating. If anyone of you want to do with me, so what you have to do, just type in a Google PubMed. Click on the PubMed, right? Click on PubMed, and then it will open like this. Then go to the mesh database. Click on mesh. Click on mesh. So it will be open like this. Mesh database. Mesh is your medical subject heading. So for examples, so you can take any research question. Recently, we are working on one research question related to non-alcoholic fatty liver disease, NAFLD. So I am just putting that. Everyone open this. NAFLD. And now I am selecting this NAFLD from here. My research question is, for example, role of bile acids in NAFLD. This is a research question recently we are working. So if you scroll down, you can see that in down, these are the entry terms. If I am including the mesh terms NAFLD, so these all terms are going to be included. NAFLD, these non-alcoholic steatohepatitis. So what I have to do, I will add it to the search builder. So I have added it in the search builder. So above you can see there is an option add to search builder. So I have added it into the search builder, right? Now I will go back. Now my next mesh term is means my I want to see the effect of bile acids in NFLD. So my next term is bile acids. So I am putting bile acids, right? Now I am putting bile acids. Now you can see that there is a bile acid salt. There are particular bile acids as well. So I am just selecting first one. If you are interested for all, then you have to select second, third, like that. For example, I want to include this bile acid as well, so I have to include, right? But here, important thing is when I will add to the search builder, if I am using and, then I will do the mistake. I have to be used from here or. Are you getting my point? Or boolean operator I have to use because bile acid salt, or because I want to include both of them, right? So I am using or instead of and then add to search builder and search PubMed, right? So on PubMed, with respect to if I am adding non-alcoholic fatty liver disease, these many 30,873 articles are there with respect to non-alcoholic fatty liver disease, right? Now I am going back. If I am adding this, next is bile acids, and this another bile acids I have added in the search builders. I hope it's not too fast. Can anyone confirm if it's too fast? then I will repeat it again. So I will ask every one of you to do it because it's a simple exercise which you can learn how to do the means make the search strategies in the PubMed and how to use for the systematic literature review. So now I am clicking on search. So these are the articles which are related to bile acids in the PubMed database, that is 43,853, 43,853, right? So now when you click on the advanced, you can see that these are the you just click on the advanced, you will find that these many articles are related to non-alcoholic fatty liver disease. These many articles are related to particular bile acids, right? Now my research question is what? My research question is not to see the articles individually on non-alcoholic fatty liver disease, or my research objective is not only to see the articles related to bile acid only. My objective is to see the role of bile acids in non-alcoholic fatty liver disease. So to do that research questions, what I have to do, I have to combine this first and second one, right? So how you can combine it? Copy it from here. First in the query box you can do it first. Now I have to use boolean operator and instead of or, and, right? Because I want to see only those articles where bile acid and non-alcoholic fatty liver is there. So I am combining this with 1 and 2. And now I am clicking on search. Are you getting my point? Now I click on search. Now only those articles will be reflected out where bile acid is tested in the MSLD. Now you can see that number is reduced 695. Otherwise, if you go to the advanced, so there is 43,000, 30,000. Now, according to my research questions, article has been reduced. Now, article is 695 only. Are you getting my point? Anyone has any question on this? किसी का कोई क्वेश्चन है इस पे? Let me see if anyone is putting in the chat box. Sir, what is the boolean operator? Boolean operator is अजय and or. जो और लगाया ना मैंने वर्ड के बीच, और बाइल एसिड और एंड नॉट इज आल्सो द बूलियन ऑपरेटर, बट नॉट वी आर नॉट यूजिंग. जो आप टर्म्स के बीच, जैसे आपको लिटरेचर चाहिए, यू वांट टू सी लिटरेचर्स वेयर बाइल एसिड टेस्टेड इन एमएसएलडी, तो बाइल एसिड के बीच में आपको एंड लगाना पड़ेगा, देन दैट लिटरेचर विल बी कम आउट. इफ यू आर यूजिंग और, देन 74,000 studies are there. You can see that, right? So that is the difference between these two. Voice is not coming. Okay. इंटरनेट कई बार बीच में वो हो जाता है. Am I audible? Voice is not audible. Okay. Now am I audible? Can anyone put in the am I audible? Now it's open for your questions as well or queries. Means you can put in the chat box, or we can move ahead. Okay, now I am audible. Right. Great. So we are talking about these PubMed search items, how to do the search in the PubMed. So I hope now you get an ideas. You have to use proper mesh terms and Boolean operators. You have to combine your mesh terms with the Boolean operator. Like if your objective is, you can see on the screen, Gapiron or MDD, means your objective is to see the effect of Gapiron in MDD. So you can't use or. This is the Boolean operator. Someone is asking in the chat box, what is the Boolean operator? This is the Boolean operator or, जो आप दो वर्ड्स के बीच में लगा रहे हो. So you can't use or, you have to use and. If you are using or, so you can see that 20,000 means by this one all the article will be picked up where Gapiron is mentioned, where MDD is mentioned, right? So on the search strategies, we have recorded separate video as well. If anyone want to get more detail how to do the search, how to make the search strategies, you can see that videos as well, right? So all these mesh terms, and then next step is you have to be, next thing is now you have to start the screening of the articles. So how to do the screening of the articles, and that we call first pass screening. So first pass screening is based on the title, without opening any articles. You just, here is the square box. In this square box you can click, it will be selected, right? So if for example you just read the title, there is no need to open the full text, means full text or abstract of the articles. So this is be called first pass screening, without opening any abstract anything. You just read the title and you feel that yes, as per my aim and objective of the SLR or meta analysis, this article is important, just select it, click here. If second article you are finding that no, it's not important, means this is not article related to my research questions, leave it blank. So like that, this exercise you have to do, you have to select from here, and finally you can generate the abstract of those articles. Here option is there in the format, click on the abstract, create file. So abstract of those articles will be created automatically, which you have selected according to your objectives. Are you getting my point? I can show you. Let me see. Let me try if I can show you on the. I hope my. Can anyone confirm it? Is my Google page is visual? Can anyone put in the chat box? So in the PubMed, right? So for example, I am just putting any name. For example, ciprofloxacin. Just showing you means how it works, right? Right. These many articles are there now. I am doing first pass screening, right? So first pass screening, I have, there is no need to open the articles. Just read the title. And for example, this article is important as per my aim objective. I will click here. You can see that in the square box, it will be selected, right? Second article, I feel it's not important, I left it. Fourth, by reading the title, I feel it's important for me. This is important for me. This one is not. So like that, you have to do the screening. This is called first pass screening based on the title, right? And finally, you can do, here is the options, save, click on save. Here is the format, select. You can create the Excel file as well, you can create the abstract file as well. So create file. So what will happen by these options, the article which you have selected, their abstract will be automatically you will get it. I don't know whether it work or not. Let's see. Again buffering. Yeah, it's very difficult to do this. How I will train you. Anyone has any questions? Am I audible? Can anyone confirm it? I hope now this voice, my voice is audible. I can stop sharing. Now you can put your questions. Rupinder Kaur, do you have any questions? It's back now. Yeah, means in other days our internet speed is too good, but when we are particularly organizing some online meetings or something like this, that day your internet will be fluctuate. I don't know why it's like that, but I am very sorry to all of you. One thing is, as I said, means there are so many. I never expect that so many of you want to learn these techniques. So I thought of maybe very few of you will be joined because weekend is there. So maybe only less than 100 will join. So that's why a meeting is organized by the Google Meet. But what happened actually, after 100, you can't add more participants. So that issues we have faced today is, that's why it's done on YouTube. So that the means many call, many messages I got on this my number, and I am the alone to handle all these. Next time I have to be keep few students with me, so that they can handle it better. So again, means we all are in learning phase, and we can learn the things by doing the mistakes. So that is a one of the things which happens, which I again never expect that. But I hope something you got it. But tomorrow's what we can do is, we can do only the YouTube live sessions. And when, because when I am doing with both Google Meet and YouTube, so it's also little bit, my system is also hanging in between. So that is also in Zoom meeting also, someone is putting, what about Zoom meeting I also checked. And so from last before 11, I am struggling how to do this. So I checked with the Zoom as well. Not many people, you can't yes, yes, with Zoom, what is the problem is, it meeting will be for 40 minutes only. Be kind is putting message, be kind, in Zoom meeting, what is the problem is, your meeting will be automatically disconnected after 40 minutes. But yes, you might add more than 100, but your meeting will be attended in means ended in 40 minutes. It's only for the 40 minutes. Then again you have to send fresh link, then again you have to send the fresh link. So, anyone have any solutions for this? They can put means give me the message. But I didn't get any solution. So only solution is this one only. Of course, I can't hear you, but I can see your comment in the chat box, and I will take your each and every queries. That I can promise you. Whatever the questions you have, I can't be hear you. Means what, I can take your each and every queries, right? So I don't want that your time will be, you feel that your time will be wasted or like that. So means whatever I knows, I will try to communicate to all of you, whether you are new to SLR metas or you have already done. So what kind of mistakes people are doing and how to deal with these mistakes. So that is the purpose, and so our country's students should have the expertise in these kind of techniques. And the major advantage of this technique is, there is there is no lab chemical is required, nothing is required. You need the skill, but you should know the proper method, proper methodologies. You should not go in the wrong directions. That is important. And you can do lot of good meta analysis or all these things in a very good impact journal. Even you can submit your result to the ICMR, to the policymakers. In ICMR, there is a Department of Health Research. You might, every one of you might know about DHR. So in DHR, they are giving the projects on SLR and meta analysis as well. They are giving the these kind of projects. So you keep on watch if there is a faculty members who want to apply projects for SLR. But of course, to get these kind of projects, you should have good publications in your profile. Then only you can get such kind of projects. But this is also one of the very important areas where all of you can do very good in this particular area. But important thing is, you have to pick up important topics, preliminary searches. You have to be very vigilant. If anyone has already done, so it will not be work out. You will face the problems in later phases. And another thing is, these SLR and meta takes the time. you know Even though now we have some AI based tool which will help us to do the screening faster, but still it will take the time. Sometime, student think of, okay, I am doing animal experimentations, I am doing in vitro studies, simultaneously I will do one SLR and meta as well. Yes, you can do it, but it will take time, right? It will take time, means when you are pulling out the data, and data should be cross checked and each and everything. And you have to be very careful, because you are doing the analysis of the already published data in the literature. So if you are skipping the wrong number, so it will hamper, right? Missed a major part of these sessions. Yeah, sure. I think someone, Asuma is putting, kindly share the recorded video for some of us. Yeah, sure. I will do that part. I will share. And sir, regarding these search strategies, and particularly if you see in this particular YouTube, we have recorded and put it some session on that one as well. You will get that. I will share the link. I can share you with now as well. Otherwise, I will forget, and you will also forget, right? So another thing is, with respect to SLR meta, as meantime, anyone has any questions, means anything which comes in your mind. What actually, sir, what is the purpose of these techniques, why we want to do it, why this so extensive exercise? As I said, two independent researchers should screen the data. Whatever I have shown you, it should be done same thing by the two independent researchers. Now, question should arise in your mind why it is so. The reason is because, for example, I am doing the screening. According to me, maybe this literature is important. According to you, maybe it's not important. So we don't want to miss anything. So whatever, means there can be a disagreement between the two peoples. According to one researcher, this is important. According to another researcher, same research question, they are saying no, it's not important, and they have their own justification for it. So now who will resolve this conflict? So that's why there is a third authors, third experts, or with other experts, you have to discuss this disagreement, and these things we have to mention in our protocols, or in who is going to resolve the disagreement. And if you see the writing of systematic literature review and meta-analysis in methodology sections, you will find that even we are giving the surname of the authors. Like if this particular authors, like for example Subham Kumar, so SK, and any other name, right? Arun Kumar AK has done the primary screening. Then third author, this surname, has resolved the disagreement between them. And you have to also mention how many disagreements are there. And if you are getting lot of disagreement, so what does it mean? It means that your research questions are not defined, means not clear in the screening researcher. Because if you are getting lot of course few disagreement is okay, but if you are getting lot of disagreement, means you have done screening of 100 article, and you are getting disagreements between 70 or 80 articles, that means research questions are not clear among the researcher. And if research questions are not clear among the researcher, then there is a lot of disagreement, right there, because research question is not clear, so how their screening will be aligned up? So that are the practical challenges which you can face. Regarding this, I can share in the chat box regarding this one as well, risk of bias assessment search strategies, right? So I can share this one as well. So I think meantime our connection is also not that much good. So maybe if you have missed out, so I have put it the link in the chat box, which will explain you about the how to make effective search strategies in the PubMed, right? In particular, I think maybe in somewhere video is not working or something like that. So in this video you will find that how to make effective search strategies by combining mesh terms and boolean operators, right? And our, as I said, our search strategy should be like that. We should not want to miss any articles, right? Which is important. Use a pre, yes, Ajay Singh is saying use a predefined protocol. That's why, Ajay, we said, I emphasize that when you are planning for SLR meta analysis, you should have a predefined protocol, right? You should have it, and in protocol you have to mention the search strategies as well. And these search strategies we also putting in the supplementary file. This is the search strategy which we have used, right? Put in the chat box. Anyone have any questions? Yeah, Lens Asuma. I have shared this one, right? This one is particularly for the search strategies, this link, right? So you can go through that link for the search strategies. Risk of bias assessment also, there is a recorded videos which will help you. We can discuss this in tomorrow session as well, but before tomorrow you can go through risk of bias assessment as well. So risk of bias assessment, after doing this exercise, first pass screening based on title, then based on abstract, then finally you get to know that these many full text articles I have to be include for this particular meta analysis. Now you can, when you have a full text article, you can do the risk of bias assessment, right? Sir, what is meta-analysis? Considered a research article or review article? Research article only, Nandini Gupta, research articles. That's why in exclusion criteria of the meta-analysis, we have to mention review article we are going to exclude. Review article, narrative review, already published systematic literature review, meta-analysis, we are going to put in the exclusion criteria. We are going to include only the research article. Why? Because from the research article, we will get the data and that we can analyze, and that is the meta-analysis. Right, Nandini? Is it clear? Any other questions? Yeah. Sir, we have published meta-analysis as a review. Yeah, some articles are, considering it as a review because it's a systematic literature review. Some journals are putting a different sections like meta-analysis section. And even some journal are considering it as a research articles. So it depending upon the journal. We have published in various journal. One journal has considered it as a research article. One journal has put it our another meta-analysis as a meta-analysis, means they have mentioned above this meta-analysis, whereas one journal has mentioned review. So journals are taking it accordingly themselves. So some are taking it a research, some are taking it a review. Got it? Yes. Next questions. Any other questions? You can put means so any question. Don't worry about any silly questions because if you are in the starting phase, so I can understand. In starting phase, I also means have so many questions. क्या है ये सिस्टमेटिक रिव्यू, नैरेटिव रिव्यू क्या होता है, इसमें क्या होता है, उसमें क्या होता है. So that all things comes in the mind. There is no issue, right? And also we have written one book as well on meta-analysis with Springer, meta-analysis in clinical research principle and practices. But of course for Indian students it might be costly, so I am not means recommending that book, but I am just putting in your knowledge. There is a meta-analysis book is written. I can share you, but even though publishers are not providing us the soft copy, otherwise I will share the soft copy, because they knows if they are sharing the soft copy, so we will share among each other, so no one will purchase it. But okay, fine. So that is a book link I have shared, right? Meta-analysis in clinical research. Any other questions anyone have? I hope now my voice is clear. Can anyone confirm it? My voice is clear, because since morning I am also getting headache because due to this zigzag, means from Google Meet to this, then I stuck somewhere as well. Then I run fast and then reach. Then there is a mismatch as well. Yeah. Okay. Great, Ajay. Any other questions? Anyone have? Anyone? Any questions you can put if there is anyone online? Okay. Sure. Okay, it's clear. Okay, great. Asuma audible. Okay, great. Yeah, one more question, sir. We can do our research while writing the research article. Can we add the data from the review article? Sir, we do our research while writing the research articles. Can we add the data from the review articles? No, means if you are doing the some research, if I understand your question, so you can't add the data from the review articles in your research. That will be the highly discouraged, and you can't do that. Means if your question, if I understand better, you are saying that if you are writing any articles, can you pull out the research data from other articles? No, you can't do that. Of course, if you want to pull the research data for the analysis, that comes under meta-analysis. So that is the meta-analysis where We are actually doing that part. Means we are pulling the data from the already published research article in the literature, putting in the excels, then combinedly we are analyzing it. Like, so, for example, RCTs, for example, on particular interventions, there are 5 RCT has been done on bile acid and MSLD. So I can pull out the data from all these 5 RCTs, and then I will make the conclusion out of it. Maybe out of 5, one article is saying it's effective, another is saying it's not, another result is significant, another one is non-significant. So I will pool that, and that is a meta-analysis. I will calculate the overall estimate measures, and then I will conclude that, okay, combinedly whether it is a significant or non-significant. Are you getting it? Be kind, do you got it, my point? Any other questions? Anyone has any questions, anything comes in your mind with respect to SLR and meta analysis? Okay, got it. Okay, thank you. Anyone has any questions? I will wait for a few minutes more, or we can, you know I think we can go ahead. Little bit I can tell you about the risk of bias assessment, right? Because it's already only we have 30 minutes, so why did you choose meta-analysis over why did you choose a meta-analysis over a systematic review? The systematic review and meta-analysis are means combined part. As I said, Agnesha, means to do the meta-analysis first you have to do the systematic literature review. Without systematic literature review you can't do the meta-analysis. Are you getting my point? So your question is, why did you choose a meta-analysis over a systematic review? Means meta-analysis. Why people are means interested for that? Because you will get some graphs or all these analysis, so that it will be a little bit easier to publish because you have done all these analysis. But as I said, to do the meta-analysis, if any one of you are planning for a meta-analysis, first you have to do the systematic literature review, then only you can go for the meta-analysis. Because what is meta-analysis? Meta-analysis is the analysis किस चीज की होगी? Analysis of the data from where you got data which is already published in the literature in the form of research article. In the form of research articles, there are so many research articles in the database. That's why we have to define inclusion and exclusion criteria as per our research question. हमें दूसरे इतने सारे लिटरेचर से मतलब नहीं है. लिटरेचर तो इतना मिलियंस ऑफ लिटरेचर है, रिसर्च आर्टिकल भी बात करें डाटाबेस में. So particularly I want only those articles which are relevant to me, which is relevant to my research questions. That's why we have to do the search strategies according to that. Are you getting my point? For precise effect sizes or subgroup analysis, yeah, subgroup analysis we are doing, particularly, you have a good number of studies, and now you want to see what is the effect in particular populations, what is the effect of these particular interventions in particular age group, particular gender. So that is the subgroup analysis. It depends upon, again, what kind of means how much literature is available, and then you can do the segregations, right? Any other questions? We can take few more questions as well. Any question you put in the chat box. I open the chat box and I am waiting for your questions. And questions are very important to ask. कुछ भी पूछो, but पूछो. I am sharing the screen. Is my screen is visible? तो अगर क्विकली मैं बात करूं, क्या क्या हमने अब तक किया, what we have discussed so far. We have discussed इंट्रोडक्शन हमने कवर की. फटाफट मैं आपको वो कर देता हूँ. ये हमने बात की थी कि क्या स्किल चाहिए हैं एक पर्टिकुलर, अगर आपको अच्छा सिस्टमेटिक रिव्यू एक्सपर्ट बनना है तो क्या स्किल चाहिए. फिर हमने बात की थी कि आपके जो ये हैं पर्टिकुलरली रिव्यूज़ किस तरह कितनी तरह के होते हैं, सिस्टमैटिक रिव्यू की हम बात कर रहे हैं, तो एसएलआर क्या होता है, एसएलआर के क्या पॉइंट्स हैं, मेटा एनालिसिस क्या होती है, वो मेटा एनालिसिस में हम पर्टिकुलरली करते क्या हैं, व्हाट इज़ द डिफरेंस बिटवीन सिस्टमैटिक रिव्यू एंड मेटा एनालिसिस। अगर हम हेल्थ रिसर्च पिरामिड की बात करें तो मेटा एनालिसिस को कहाँ पे रखा गया है, मेटा एनालिसिस एट द टॉप because that guide the clinical practice guidelines as well. So when it is required, we have covered, means when there is a confusion among the physicians, how to select the topic for the SLR meta-analysis. So we have discussed conflicting evidence, so policy implications, so ongoing scientific arguments. So then you have to check, then you have to be go through the prisma guidelines. So prisma is the preferred reporting items for the systematic review and meta-analysis. So recent, you have to go through, and recent checklist and flow diagrams you have to download, right? And there is no need to create figures or no need to create the checklist is available, you have to just fill it. So you have to fill it when you have completed your SLR and meta analysis, then only. And this is the flow diagrams, so again I am repeating, there is no need to create a flow diagram by yourself. There is a flow diagram which is available on the website, download it and put the numbers. और इसमें वही नंबर भरे जाएंगे जैसे इनिशियली सर्च हाउ मेनी आर्टिकल्स यू गॉट, then after first pass screening how many articles has been removed, how many articles has been included. So we are creating a excel file for the excluded articles as well. And then finally how many articles you have included for the qualitative assessments, how many articles you have included for the quantitative analysis, that is your SLR, sorry, meta analysis. Then we have discussed about the protocol development is important. Then you have to register this protocol in international registry like Prospero में आप सब अपना account बना लो, and then put the for the registrations. Once you got the registration number, you can put in the, and ICMR as per the ethical guideline, there is no need to do that. Means no need to put in the ethics committee and involve a team. Important points जो हैं कि आपके clearly formulated research questions should be there, your research strategy should be comprehensive, transparent inclusion exclusion criteria, quality assessment, data extractions. Then we have discussed the steps. So these are the major steps which you have to take. First steps, I hope every one of you are clear about the first step which you have to take, how to search the literature in the database. I have shared the link in the chat box as well for the search strategies, how to make the search strategies. And then so we are doing the eligibility of the studies, which studies you have to include. As I already said, case report, case series, review articles, we have to be excluded from the meta analysis. So ROB assessments, when we talk about risk of bias assessment, risk of bias assessment depends upon the study design of the included studies. Depending upon the study design, you have to be pick up which tool is important for this particular risk of bias assessment. Like Cochrane risk of bias 1 2008, and now it's revised in 2019, that is a Cochrane ROB2, is used to assess the to do the risk of bias assessment of the RCT, randomized control trial. If your study design is randomized control trial, you picked up this tool for the ROB. If your study is non-randomized clinical trials, then you can select the Robbins one, right? If your studies are observational, you can pick up Newcastle Ottawa scale. If your cross-sectional survey, you can pick up Access. If you are dealing with the diagnostic test accuracies, systematic review, you can pick up QDAS. If you are dealing with the animal experimentation, you picked up S by R CLE, right? So depending upon the design of the studies, you have to be picked up risk of bias too, right? Are you getting my point? So this is the Cochrane risk of bias assessment. Scale is means tool is there. So how to do it? So you have to consider all these point. So random sequence generations means you have to go through the individual. This assessment is done based on the individual articles. So in article, if randomization is mentioned, so you have to be select low risk. If it's not mentioned, you have to mention high risk. If it's not clear, you have to pick it up unclear risk. This is the recommend, right? Allocation concealment, selection bias. Allocation concealment is mentioned or not. So all these points you have to be check in the articles, and if it's mentions, then it comes under low risk. If it's not mentions, then it's a high risk. If it's not clear, you have to select the unclear risk, right? So I'm just giving the glimpse that we can discuss in detail tomorrow as well. Then risk of bias assessment, what are the critical appraisal, then there is a extraction of data in the data extraction sheet. So that you have to be designed according to your research questions. We have to be design the data extraction sheet. That is very important, because on the basis of data extraction sheet we have to do the analysis of the data. And tomorrow we will discuss how, what is odd ratio relative risk, and when we will go with the odd ratio relative risk, or mean difference or standard mean difference, what is variance with 95% confidence interval, and so on. And what is the plots, forest plot, funnel plot, how to do the analysis. So all these points we will discuss tomorrow. How to select the models, how to deal with the heterogeneity, what is the I-wall test, what is the chi-square test, I-square test, how to deal with that, publication bias analysis, how to handle subgroup analysis, and what are the AI based tool which can help in these processes. So all these we can discuss tomorrow, right? Meantime, any questions, I am happy to answer. We have 5-10 minutes. Yes. Yes, there are 2-3 questions. Sir, what mistake do beginner make when they are just starting with SLR and Meta? Kajal Sharma is asking good questions. What mistake do beginner do is, one mistake beginner is doing is picking up the topic. Means selection of topic is very important. If you are picking up topic, means not a good topic for SLR Meta, then you will get the difficulties. Your the work will be criticized by the experts as well. Why you have done it? What is the advantage of it? What is the output? Right? Everyone is nowadays asking about, if you are doing any exercise, anything, what is the benefit of it? Right? That question always should be in the beginner's mind. I think today's generations always put the question, "Is sir iska kya फायदा होगा इससे, what is the output, what is the benefit?" So that question should be clear. Okay, this is the benefit, this results will guide the clinicians, because this is a conflicting evidence. So literature, and sometime, as I said, means beginners, what they do, they picked up the topic, they are not doing the preliminary search as well. Preliminary search means you are doing random search in the databases, and just to check whatever the topic I have picked up, whether someone has already done it or not done it. If someone has already done it, so what they have done and what I have planned, what is the difference between these two? How I will defend if someone asks me, this person has already done this, so why you have done it again? Right? So that is the another important points for the beginners. They are doing the mistakes there. They are not checking up, they are not doing the preliminary search, and last they are not getting anything out of it. Another thing which beginners are doing is, they are doing the mistake is, they are not writing the protocol. They are just started the work, they are started the SLR work, and doing the screening without registration of the protocol, without writing of the protocol, without the registration of the protocol. So first you write the protocol. By writing the protocol, you will be clear about your research questions, so you will do the screening of the studies in a better way. Are you getting my point, Kajal? So these are the some of the mistakes. And another mistakes which beginner is doing is, they are not able to make proper search strategies in the databases. They are doing random search. Random search is not acceptable in a systematic literature review. Your expert will ask you how you have searched it, what is your search strategies, which mesh terms you have used, from where you got it, which boolean operator you have combined. All these things, beginners are doing the mistake at this point, and these points are very crucial for any SLR or meta analysis, right? So just starting with SLR meta, be clear about these points. And every one of you can do it. It's not very hard task, means everyone, but you have to be very careful when you are doing these kind of work. Next question is, I hope, okay, Kajal, is it clear, right? Agnesha question is, how to address high heterogeneity? Yeah, you know, s- heterogeneity is something Meta-analysis are always criticized for mixing apple with oranges. You know why? Because your research question is defined, but you are pulling out the data from the different studies. Different studies. Now you will ask me, sir, I am taking only the RCT. Okay, you are taking only the RCT, but just compare the RCT. There is a differences among the RCTs as well. For the same medicines, one company has done the trial on, their age group is 45 to 50. Another company is doing to 60 to 65. Another, their baseline are different. There are variations, right? So obviously you will get the heterogeneities, but the question is how to address it. So how to address is, we have to go for the meta regressions, or we have to go for the subgroup analysis. So we have to check the reason for the heterogeneity. Why am I am getting 100% heterogeneity? What is the reason for it? Right, which study is mostly contributing for the heterogeneity? So you are doing the subgroup analysis. You are doing the subgroup analysis based on the strength, right? Initially, for example, you have done the analysis based on all the doses, all the strengths. So now you have to do the subgroup analysis. Okay, I am keeping 20 mg's intervention different, 40 mg different. I am doing analysis differently for the 20 mg, differently for the 40 mg. Now I will check how much heterogeneity has been reduced. So you have to be, find out the reason for the heterogeneities and to deal with that. And still, if you are getting very high heterogeneity, then you have to put in the limitation sections that there is a high heterogeneities. So means these results are, you can't give the clinical recommendations for based on that. I hope it's Agnesha. That is a meta regression. You can read about meta regression to deal with heterogeneities. Next question is Ajay Singh. Firstly, identify source and then apply appropriate statistical and mathematical corrections. Yeah, means firstly you have to identify the source, means databases. Once you pull out the data in the data extraction sheet, then only you can analyze it, right? Any other questions? Any other? Still 4 5 minutes I have. Anyone has any doubt, anything they want to say, you know, they can put. Okay, so tomorrow's sessions will be, if you are comfortable with this mode, so then we can meet tomorrow. If something today is useful to you, then only I can continue tomorrow. Otherwise, we can postpone it as well, because we are not able to accommodate more than 100, and many are in waiting. So that will be very difficult. So let's see if you are planning to join tomorrow's as well. So tomorrow we can cover the, so next step is we can spend some times on the designing of data extraction sheet, and then we can go for the analysis of the data as well, how to analyze. And of course, one more important part is grade analysis, grading of evidence. Now we are talking about grading of evidence. So that also we can, means I will try to touch on grade analysis, how to do that after completion of your meta analysis. We are grading the evidence, because based on the meta analysis results, you can give the recommendations to the policymakers, to the clinical practices, to the researcher. Now you can understand that how important these kind of analyses are. You are giving recommendations to the policymakers, to the clinicians, to the researcher. So that's why grading is very important. Depending upon the grading of the evidence, your recommendation will be decided. You can't recommend anything to the policymakers. So you have to do all the assessments by the grade, and then you can give the recommendations to the policymaker. So grading का मतलब क्या होता है? In school times you are getting the grade A plus, B plus, B. So based on what? Based on what, means what performance you have given in your exam, or based on that and certain parameters. So like that, in this case, certain parameter is based on the included studies. So included studies, we are doing the grading. Okay, but is the biasness, which biasness, indirectness, impactness, all these parameters we consider, and then we are doing the grading. And based on the grading, we are giving the recommendations. Okay, this is the high grade results. So based on that, you can implement in the policy. Okay, this is means not that much, yeah, but we can give the recommendation to the researcher to do more research in that particular area. So like that, that is a grade. So grade, nowadays everyone is talking about grade. If you go through the ICMR websites, they are conducting workshops or webinar as well on the grade analysis. Now earlier we are talking about SLR and meta analysis only. Now we are talking about SLR, meta and grade analysis, right? Okay, okay, then if there is no questions, no more questions, so grade का भी कर लेंगे, there is no issue. So thank you so much everyone for your time. Have a great day. Have a great Saturday. Thank you. Thank you everyone. Thank you.